The overall goal of this procedure is to study virus assembly in vivo and in vitro. This is accomplished by first expressing brome mosaic virus RNAs in bacteria and exposing it to plant cells through agroinfiltration. Next, the BMV virions are purified from infiltrated leaves. Then, capsid protein subunits for in vitro assembly are prepared. Finally, BMV virions are assembled in vitro using the capsid protein and indocyanine green dye. Ultimately, the results show in vitro assembled brome mosaic virions through formation of optical ghosts, which can be determined by absorbance and emission of fluorescence.
So today we are going to show a technique which is referred as agroinfiltration, and this technique is useful for delivering viral components to the plants. And the main advantage of this technique is to eliminate making in vitro transcripts. And at the same time, it delivers multiple components to the same cell, which permits a synchronized infection.
The work that we are currently pursuing is the use of the brome mosaic virus.